Hi, I'm Sharon Anderson, president of Leiomyosarcoma Support and Direct Research Foundation, or LMSDR for short. And I'm really excited today to have our guest speaker, Barbara Van Hare from the Rare Cancer Research Institute. And reason why is because one of our missions is to build a bridge between patients and researchers and to work for a cure, not wish for a cure. And Barbara's going to tell you, yes, you, how you can participate in research. So, Barbara, you want to take it away?
Sure. I'm going to pull up my slides as I introduce myself. I am Barbara Van Hare, and I am with the Rare Cancer Research Foundation, and my actual title is President of Pattern.org. And I'm going to put this to full screen. Can everyone see that?
Yes.
So what I'm going to talk to you about today is the power of fresh tissue donation through Pattern.org. And the Rare Cancer Research Foundation is a 501c3 nonprofit that was founded by a rare cancer patient who wanted to break through the barriers in order to expedite research for not only his rare cancer, but for every rare cancer. And so one of our key initiatives is we launched Pattern.org in 2017 to enable patients to directly donate their fresh living tumor tissue and medical data to high impact research. And so therefore, through Pattern.org, we essentially bridge the gap between patients and scientists and are empowering patients like yourselves to have a critical role in helping to improve the outcomes. So you as patients and caregivers who can help communicate to patients have the power to make a difference. See if I can get it to go to the next slide. So you may or may not be aware on how important tissue donation is, but particularly with rare cancers, by the fact that they're rare, there are limited patients at any single institution. Even the largest academic centers that study cancer, like MD Anderson, don't have enough rare cancer patients, and so they need other ways to access tissue in order to do research. Many institutions store tissue, a lot of institutions don't, but when tissue is stored, it's incredible when there are research projects at that institution that are going to use that tissue. But in many cases, that stored tissue stays at the institution in a biobank and never sees the light of day. As I mentioned, researchers don't have access to tumor tissue because in the nature for rare cancer, it's rare. And because researchers haven't had access to tissue and traditionally pharma hasn't funded a lot of rare cancer research, there aren't many robust model systems for rare cancers like LMS. And what I mean by a model system are things like cell lines or organoids, which are actually models that researchers can learn from and test drugs against. And then finally, lack of open source models and data. The way research is structured today, researchers are kind of incented to hold back on their learnings until they actually publish a paper. And it's only after they publish that they'll share a lot of information. And quite frankly, in order to get something published, it can take not only months, but years. And in some cases, things are never published. So patients can become partners in helping research through tissue donation to help solve these issues. And particularly fresh tissue. And what I mean by fresh tissue is tissue that is taken at time of surgery. So you have a tumor, and when the surgeon is removing the tissue, they can take a piece of it or they can send it to pathology directly after the procedure. And some excess tissue can be taken from that in order to be sent to researchers so that they can better understand the biology of the disease, like what is causing the disease. They can test and identify ways to help diagnose and hopefully prevent the disease, and also test and identify potential new therapies and cures. And so how does Pattern.org work? So a patient would go to Pattern.org and they can learn about different research studies for their disease. The patient would then put in some basic information such as their name and what disease they've been diagnosed with. And then they are matched to a study that's in need of that fresh tissue. And then the patient can go through the different screens online and consent to give some excess tissue from their procedure to research. Once a patient consents, literally our team is notified within about 60 seconds. And then Pattern will reach out to the clinician at that patient's hospital, talk to them about the patient's request to donate excess tissue to a particular study. Then we work to get agreement from that institution. We send in a kit, and I'll show you a few details about that, so that at time of surgery, they can put some tissue in that kit, and then we will pay to overnight it to that research project that the patient was matched to. So on the left-hand side here is a picture of what one of the kits looks like. It comes with what's called a nanocool, which makes it easy for the institution that's doing the surgery to just press a button so it basically stays at a refrigerated temperature. Again, this is fresh tissue, not frozen tissue, so we need it to stay so it doesn't get too cold or too hot. On the right-hand side are basically similar to the tubes that the tissue would go in. And normally we ask for the doctor to give us a little bit of tumor tissue, some adjacent normal tissue if it's available, and or blood. And the reason for the normal and or blood is the researchers, they'll have the tumor tissue, but they'll want to compare it to what's considered to be normal tissue or non-tumor tissue. And so through either the normal tissue or the blood, they can compare what's normal for that individual versus what potentially is different about the tumor tissue. As I mentioned, patients are the ones that consent online, and literally we can work with any hospital or medical system across the US. Since we've started Pattern a few years ago, we have collected fresh tissue from 123 different hospitals across the US, and they have been represented across 39 states. And so many people ask, so explain what's the difference between what pattern.org does and what the researcher does. So pattern.org helps break down the barriers in the infrastructure, i.e., we're the ones that help reach out to patients through foundations like Sharon runs. We're the ones that have the pattern.org website where the patient can go on and learn about these studies and consent. We're the ones that will do the outreach to the hospitals and then send in the kit and then overnight the kit with the tissue to the research study. And we're the ones that work with the research studies up front to make sure what they're doing with the tissue is covered by our consent and protocol, because we are overseen by a regulatory body called WCG-IRB. Now, what the partner research studies do is they do all the scientific work. They're the ones like trying to develop these living models. They may do genomic studies. Almost all of them do genomic studies. They may do what's called CRISPR screens to learn more about what's different about this particular tissue. They may do drug tests, and they're the ones that potentially publish papers about, about the tissue and results, the models. And then in order to work with pattern.org, these labs agree upfront that they will share all of the information that they gain from these tissues. So whether they create models or genomic analysis, any data they generate, all of it's de-identified. When we send the tumor tissue and any information about the clinical history of the patient, the research lab does not know who that patient is. And then when they share the results broadly, that is also all de-identified, so no one knows who that patient is. And that's where the work we're doing is a bit different, besides the fact going patient direct and working with institutions across the country. We require that they can't keep the results siloed within their institution. And that's so that the broad research community, meaning researchers around the world, have the ability to look at the data and access the models so that the research can continue faster than it has been historically done. I know a lot of LMS patients have contributed to the Count Me In project, which we kind of consider to be a sister project to ours. It's a different group, different project, but we're very friendly with one another. Where Count Me In, they really want stored tissue. So tissue that is already at the institution that is stored. And they are doing their studies primarily to generate genomic data, and they will share that broadly. Where we're a little different is we're going after the fresh tissue where models can be generated along with the sharing of the clinical and genomic data. But the models also allow for things like drug testing and a lot of other analytical tools. So fresh tissue is kind of the key thing that differentiates us between the Count Me In project. Again, the BRO does share the data that they generate. We ensure that the researchers share the data, and any models that they generate also have to be shared. Some key information about when you donate tissue through Pattern. We're looking for excess tissue only. So any tissue that is needed for your clinical care, if there is not enough tissue to share, that is fine. We want to make sure the patient comes first. So absolutely any tissue that's needed for patient care should stay at the institution. The sample size that we normally ask for, but we will accept less than this, we'll accept more than this, is 0.5 centimeters cubed. And we get asked a lot about biopsies. Normally, we don't take tissue from biopsies because biopsies usually generate a small sample of tissue. We can take biopsy tissue, but really surgical resections is where there's usually the excess tissue. Because we're looking for fresh tissue, patients need to consent prior to the surgery. We would ask, if possible, to have like a week's notice before the surgery. We have collected tissue when a patient consented the day before. The challenge we have if we don't have at least a few days is we have to reach out to the clinician, whether that's oncologist, surgeon, or a combination thereof. We have to be able to reach them. We have to make sure that if they need approval from their institution to release the tissue, we have to give them time to do that. In many cases, we also have to talk to the pathologist. Then we also have to get everything together to overnight the kit. And when it arrives the morning of a surgery, there's a great chance that we may not collect the tissue because the kit will go probably to whoever accepts the boxes, and then the patient may already be in surgery. So if a patient can consent in advance, that's really great. We do protect patient privacy. As I mentioned, de-identification of patient tissue and data is critical to us. We do store our data in an encrypted HIPAA-compliant data server. If, however, a patient at some point wants to revoke their consent, either before the tissue is collected or after, that is no problem. There's no cost to the patient to participate, and currently all the research studies that we are collecting for is for general research. And in layman's terms, that means we will be able to tell you that the tissue arrived to the research lab, but we are not currently, by all of the laws that govern the work we do, able to tell you exactly what came out of that tissue. That's what general research means. Our current research partners with pattern.org overall are the ones listed here. Since we have started pattern.org in 2017, over 65 models have been generated along with countless data sets, which are all critical for researchers to help find new learnings to lead to new therapies. And regarding specifically leiomyosarcoma, we have been working with LMS patients since 2017. And I mentioned that over 65 different models have been generated. 6 of those 65 have been LMS. And I am thrilled to say that all 6 of those models are at public biorepositories. One is here in the US at a place called ATCC, and one is in Germany. And what a public biorepository is a location where these models are stored so that researchers can have access to them. Researchers all over the world will be able to contact these ATCC and DSMZ to be able to access these models. The models have been sent to both of these places. They aren't quite available yet because once the models are sent, they're in basically like tubes, and they have to be what's called expanded, meaning they have to make more of that model so that as researchers order them, they have ones to replace it. So as soon as the expansion is complete on each of those models, they'll be available to researchers. In the meantime, if any researcher wanted access to them, they can contact the Broad Institute or contact us, and we'll put them in touch with the proper people at the Broad. Also, all of those 6 are undergoing CRISPR and drug screening, which is basically advanced screening to learn more about what's causing the cancer, where the cancer's what they call vulnerabilities are, so that they can better target drugs to treat the disease. All of that is being done at the Broad Institute, and hopefully in the near future, then there'll be a list of drug targets and potential lead therapeutics. On kind of a side note, but definitely related to this, one of those 6-cell models is an ultra-rare cancer called uterine myxoid leiomyosarcoma. And that model is the only publicly available uterine myxoid LMS model in the world, and it came from a patient that donated through Pattern. So that just shows the power how one patient's tumor tissue that isn't needed for that patient care can have a big impact. And you might think, oh, well, there's only one publicly available model. What does that mean? Well, what that means is about 6 months ago, we were contacted by a researcher up at Dana-Farber who is treating a patient that actually has that disease. And that particular model almost exactly matches that patient's cancer. So they're utilizing that to try to see if what the best potential therapeutics would be for that patient. So the current and future sites that we're working with that have an LMS focus, as mentioned, the Broad Institute and the Koch Institute at MIT, we've been working with both of them for a number of years, and they're the key ones that have done the work to date. Also, pattern.org and RCRF, the Rare Cancer Research Foundation, is starting another initiative called the Pattern Biobank. And basically, that is a biobank where if there's a patient that wants to donate tissue, but we don't have a current study set up to accept that tissue, we can ship the tissue, that fresh living tissue, to the biobank and keep it there temporarily until either a partner is set up, or if there's a partner that, let's say, may want 10 particular type samples before they receive them and start their study, we can store the samples there temporarily until that study is ready for it. And then currently we're onboarding 3 projects that have an interest in LMS. One is UCLA Health. There's a researcher there that's very interested in creating LMS models. The Siteman Cancer Center, which is at the Washington University in St. Louis, they have a sarcoma project where they're going to be looking at the proteins on the surface of cancer tissue. And the other is at University of Texas, San Antonio, where they want to do a research study to try to better identify how to differentiate early on the difference between fibroids and LMS. Because a lot of patients are misdiagnosed with fibroids when in fact they have LMS. And so he wants to do some research to better inform doctors early on so better treatments can come to those patients. We're also actively recruiting other partners that have an interest in LMS. So we'll have more information on these different studies as they come on board, hopefully within the next few months. In the meantime, the Broad is still doing their CRISPR and drug screens. The Koch Institute currently has enough LMS samples for the work they need, at least for right now. That may change in about 6 months. So any tissues that come to us in the next few months, we will temporarily store in the biobank until the other projects are ready. So basically, what pattern.org is about and Rare Cancer Research Foundation is empowering patients, identifying patterns in the tissue. That's where the pattern.org name comes from, is helping researchers identify the patterns that may lead to new learnings and advancing research. So on the left-hand side here in green, I've talked a bit about the live tissue donation. We hope that by early February, for patients that are interested in storing their own tissue, where we'll have an opportunity to work with those patients. There will be a cost for that to the patient, but they can store their tissue in our biobank. They have full control over it and would be able to determine wherever they wanted to send their tissue, whether it was to support research studies that we have, if it's to study research projects they're interested in, or if they need it for what's called CLIA testing. What a CLIA lab is one that's able to do testing for work that could help inform patient care. So a patient would have the ability to store their own tissue and do with it what they want, and more details will be upcoming on that. Genomic sequencing, we don't do here at our organization, but if patients are interested in genomic sequencing of their tissue and they're interested in learning more about it, we have partnerships with several different genomic sequencing companies and are aware of most all of them, and we can help patients get that sequencing so that a report is sent to their doctor to talk about what may have caused the cancer and potential clinical trials, et cetera. And then coming later next year, there'll be a patient portal with additional opportunities for patients to have access to their own data, et cetera. On the right-hand side, I've talked about the models and the data sets. We're hoping that our work will help lead to precision medicine and then the pathways to cures. The blue box in the middle is talking about our data commons, and basically we have been asked by a number of different folks at different institutions saying there was a real need for a rare cancer data commons where rare cancer data is easier to access and easier to find. So we're in the process of creating that, and that will be live sometime next year. So in a nutshell, you as patients have the power to make a difference, and we would invite you to join us to help provide a brighter future for those with LMS through tissue and/or data donation through pattern.org. Tissue is really critical, but so is data. So if for some reason you're not having a surgery or you had one in the past and you'd like to donate your medical history to us, pattern.org will be updated by early February, so you'll have the capability to do that. If anyone is interested in doing that prior to February, they can feel free to contact us directly, and we can walk them through that on a manual basis. Thank you for your time, and now I'll open it up to see if there are any questions, and hopefully I'll have the answers.
This is really exciting. I wanted to make the note that not only can you give this tissue, this valuable tissue to researchers and research projects, but you can also store it there for a fee for your own use. So I think that's really exciting. I don't think we have any other place we can do that. So I had a couple questions, but you answered them all. Did anybody else have some questions?
I do about the biopsy. So I want to understand, you need to have like a fresh tissue piece that has like a margin around it. So if
No, so when they do a biopsy, usually they're just taking a very minimal amount of tissue for basically clinical care to learn more about your cancer to determine next steps. And because we're a research project, we don't want to and can't take tissue that is needed for your care. So with a normal biopsy, occasionally biopsies will have excess tissue, but for the most part, they're just taking a minimal amount. Whereas with a surgical resection or surgery, more tissue is taken and there's usually excess from that, if that makes sense.
So I work at a hospital where we do a lot of pulmonary nodule biopsies.
Okay.
And so, and I help to make slides for the pathologist to study during the procedure to diagnose. And then we also, of course, have tissue that goes into cytology.
Uh-huh.
So it's just so it doesn't kill it it doesn't freeze it like in formaldehyde.
It's not in formalin.
Right. So often with those nodules, for example, I'm just using a nodule as an example.
That's great.
The doctor, we will get those biopsies, but then and he'll say, okay, I'm done. But there's still enough nodule that we could take more biopsy. So is that acceptable?
It is, as long as it's still living, if that makes sense. So I don't know how long, like, is it immediately after the procedure, they say that they have the extra?
Well, yeah, like we will do the biopsy, we'll get what we need. Like
Okay.
once the diagnosis has been confirmed, then we either can be done and we'll move on to something else, or he can go ahead and we can take more bites of that same
Okay.
nodule.
If that tissue that's left in the nodule is still living, so within an hour or two of when it was taken out, then
Mhm.
yes, that would work. If it's not needed for the patient care, and basically it's going to be, I'm not sure where you work, but many hospitals will destroy tissue.
Mhm.
That would be very helpful to get. And so we can send kits for biopsies. It's just a lot of biopsies. They're just taking a small amount, and there's usually not excess. But if there's excess from nodules, absolutely we can.
Okay, I just wanted to make sure that that was accurate.
That would be great.
Okay. All right.
A little more about what a model is, because they use this fresh tissue to make models.
So a model is usually refers to as like a cell line or an organoid. And an organoid is like a mini tumor. It's a three-dimensional model. It looks like just a little, I'm not going to say a bubble, but it's a kind of a little tiny tumor, I guess would be the best way to say it. And a cell line is more 2D, so it's more flat. It can be, they can, I'm not a scientist, but they could put it like in a dish, or most of the time they will save them in like a tube, similar to the tube, not identical to the tubes that we collect the tissue in. And they're like it's a living model of your disease. So it's kind of like a mini replication of the cancer that they study in the lab. It's not necessarily an exact duplication. It kind of depends, but it's one of the key things researchers need in order to learn more about the disease. Some people may have heard of something called a PDX model. That's a patient-derived cell model. It's basically when
Mouse
you put a
model.
piece of the tissue, thank you, a piece of the tissue in like an animal, like into a mouse. They're also called mouse models. And they're just a key tool to study the disease.
Right. They can test drugs on that mouse
Correct.
to see which ones work. Yeah. And the cell lines are something they continue to grow. Is that true?
So yes, a long-term cell line is one that if anyone's ever heard of Henrietta Lacks or read the book, it's an immortal cell line. And Henrietta, her cell line was created, I want to say it was a long time ago, 1950s or so, and it's still living today. That is called an immortal cell line. Sometimes as these cell lines grow, they may pick up characteristics and things like fibroblast, and so they're not long-term models. They only last a short time before other things kind of get in the way and take away what kind of made it like the original tumor. But all 6 of the cell lines that I talked about in the presentation are long-term models that should go on in perpetuity.
Great. Thank you so much. Karen, you have any questions?
I do. This one might sound a little bit morbid, but if I want to donate, should I pass? Could those, is it, should I pass from the disease? Because I would very much like to donate tissue that might help others if it's viable at the time that I pass. And so I don't know if there's a process to help make my family, my care, aware that that would be my wish.
That is a great question, and it's actually one that we're asked a lot. I can say we have tried a number of times to be successful with, and I don't mean to be morbid either, but postmortem tissue. It's very challenging because if a patient passes at home, they may not be near a hospital or near someone that's going to do an autopsy. You may have a funeral home involved. Who's going to pay for the cost? If the patient passes in the hospital, if it's on the weekend, there may not be somebody there to get the tissue right away. And after someone passes, the tissues in the body start to deteriorate fairly quickly.
Including the cancer, right? Because they need the body
So
to live.
there are some hospitals that have a program for certain cancers where they will utilize postmortem collections. We just haven't been successful with it because we work with patients across the country. We've tried.
Sure.
We did have one success. It was a very unusual situation where a very young patient, I think she was 22, had an aggressive cancer that literally was visible outside her body. And so the nurse that was treating her after she passed was able to get the tissue right away. It was kind of the patient's dying wish. But that's not normally something that we get too involved with.
So
And
what
then
else?
my original tumor was removed, and it's within Dana-Farber. It's at Count Me In. Should my cancer evolve and I need another procedure, that would be an okay time to reach out to pattern.org and say, hey, I would
Absolutely.
like this. And the interesting thing for me is there a way to compare the evolution of that disease? I mean, for one, it would help pattern, but would the original tissue in its preserved form be also a piece that should go with it so that there's ability to compare it?
We would love the opportunity. I've talked about fresh tissue donation. We also have the capability to get stored tissue. So we would love the opportunity to collect stored as well as the fresh tissue for comparisons. I'm sure the researchers would like that as well. If that were to happen, things continue to evolve. And we are the belief that the more we can help patients, the better. So what ultimately our goal is to use these tissues, yes, for research, but also to provide back information to the patient. So laws continue to change. We're continuing to do work to see if down the road from some of these, if there is information we can give back to the patients. We're talking to a startup now that has a platform where they want to create organoids, but they can't identify the patient. But I don't know all the details yet, but they're working to see if there's a way to let patients know any learnings from the research that's done with it, give the information to the researchers, but also let the patients know, but do it so that it doesn't break any of the HIPAA laws or the de-identification laws. So more to come on that. It's still early stages there, but I think you'll start seeing over the next few years more ways to get more information back to patients.
That's really exciting because I know that's a big interest for patients. And I think Count Me In will give some information back, some genetic information, and I'm not sure if they do genomic information as well.
So with The reason I mention about that we are trying to partner with a number of different genomic testing companies is we really feel every patient should have access to that and at little to no cost. And so any patient that's going to be coming through Pattern starting in the new year, we will reach out to see if they are interested in genomic testing. And if so, we can help connect to we can't recommend one company over the other, or any of the companies. There are a lot of laws with Medicare and Medicaid, but we can help connect. We can also help talk to the doctors because some doctors still don't feel genomic testing is worthwhile. And we can try to help influence. The value of it, we feel, is it may show a mutation or a something that helped cause the cancer that wasn't previously known. And I'll just give an example of our founder, who coincidentally happened to be my nephew that unfortunately passed away from metastatic disease earlier this year. He had ocular melanoma, but he had sent tissue to several different companies from different lesions he had inside his body. And the first two tissue samples he sent didn't identify a genetic alteration in his cancer. A few months lapsed and he had another lesion removed, and they sent it to a different company. And it turns out he did have a genetic component. There is a family genetic mutation that we now know he has. It turns out I have, turns out his dad has. We didn't know that before that. And it was in the report that came out. And as a result of it being in that report, I was able to be tested. My brother was able to be tested. And we now are being watched for potential cancers that come as a result of that. Turns out that information was in the data with the first two companies as well. But because it is such a rare gene that hardly anybody knows anything about it, it wasn't in the report, but it was in the data. So we also feel the data is important. So what we're asking patients that we help with the sequencing is if they're willing to share from those companies that are doing that. Some of these companies are willing to share the data with us. We can put it into our data commons to help have key learnings. And where I said like that patient portal, one of the things that will be there is patients will have access to their data as well. Like their whole, it's a large file. If a patient wants to see it or share it with others or have somebody look at it, they would have that capability to do that. So we just feel there's a lot of potential if patients are interested in getting that.
Resource for patients. I'm really excited about that. Any other questions? Is that it? We might be at the end of our.
I had questions, but now I lost them.
Well, people can always reach out to me, Barbara, so bar, and then an A at rarecancer.org or Barbara at pattern.org. They come directly to me. So you can feel free to reach out. Any of you on this call, any of you looking at this in the future, we're happy to answer questions either about what we're doing or other things that you might have questions about that if we don't know, we'll try to refer you to people that do.
Well, thank you very much, Barbara. This was really informative, and it's very exciting to see more and more research and resources for patients to access their own data and help the researchers help us. So I want to thank you. I'm going to sign off now, and we'll talk more.
Okay.
Thank you.
Thank
All
you
right,
for having
bye.
me. Bye.
Appreciate you. Thank you, Sharon.
Thank
Thank
you.
you.